one of the students looked at me as he walked by and said, "You are going to freak out," which is usually a good thing to hear before one of these meetings.
Hello and welcome to the Cheeky Science Show, where in this video I had the pleasure to interview David Liu, Professor at Harvard University, the Broad Institute and Howard Hughes Medical Institute. David's lab is using chemistry and evolution to develop next-generation therapeutics. They've developed base editors and prime editors to study and hopefully treat different genetic disorders. From his work, David has co-founded many different biotechnology companies, including Editas Medicine, Pairwise Plants, and Beam Therapeutics. So I hope you enjoyed the following interview. But first, I need to deanimate, not deaminate, as we'll learn about later. What does this do? Perfect. So hi, David. Thank you so much for joining me today, and welcome to the Cheeky Science Show. You are officially the first ever guest, and I'm particularly excited because it was probably the first video that did quite well for me on my channel was all about prime editing, which is a gene editing technique that was developed in your lab, which we'll come on to a bit later. And so it feels like I've kind of gone full circle a bit here. So it's just great to have you on.
Oh, well, I'm really honored to be your first guest, and I'm a big fan of your videos and your channel. And I think you do a great job explaining complicated science to lots of people. So thank you for doing that.
Thank you. So speaking of genome editing, what was your introduction to genome editing? Because I think most people, when they think of it nowadays, they think about CRISPR, but obviously CRISPR wasn't the first genome editing technique that was used. So what was your introduction to genome editing?
Right. So, as is often the case, the route that we follow in science is a combination of our curiosity, but also a lot of serendipity and the network of chance meetings and friends that you happen to have. So in our case, our lab has always, since it started in 1999, specialized in using sort of the components of biological evolution, including in vitro selection, mutation, replication, to address problems in biology and in therapeutic science. At the time, I had a graduate student, Vikram Patanayak, who was working on an in vitro selection system that could identify RNAs with certain biological properties. And while he was sort of thinking about what project he should do next, he happened to attend a Harvard Medical School event. He's a medical school student, was a medical school student, and happened to sit next to Keith Jun, now my good friend, collaborator, colleague, co-founder of a number of companies. And simply by the good fortune that he sat next to Keith Jun, they ended up talking about an area that Keith, was really and continues to be a leader in, namely zinc fingers. And what Vikram became interested in and confirmed with Keith that this project idea was significant was that zinc finger nucleases had already started to enter the clinic. Sangamo was running clinical trials on CCR5 cleaving zinc finger nucleases. Yet, to our knowledge, there was no unbiased, broad characterization of the DNA specificity of whole, intact zinc finger nucleases. What researchers had done as a surrogate before was to take individual zinc finger arrays and do in vitro selections against libraries of DNA to see what they bound. But you could imagine, as I think has proven to be the case for many DNA binding proteins, that the sequence requirements for nuclease cleavage are not exactly the same as the sequence requirements for DNA binding. So this led, to make a long story short, to a collaboration between our lab and Keith's lab. That was our very first gene editing. In fact, it wasn't even called gene editing then. It was called genome engineering. And then it became genome editing, and now it's called gene editing. But our very first project in that field, which was to develop and apply an in vitro selection method that could very broadly characterize the DNA cutting specificity of zinc finger nucleases in this broad, unbiased manner, where we took advantage of the fact that you can make large libraries of circles of different DNA sequences, and when they're cut, they get linearized. You can capture the linear ends. It's actually related to some of the current methods used to characterize the off-target DNA cleavage activities of CRISPR and TALENs and zinc finger nucleases. And indeed, after publishing that first paper on zinc finger nucleases with Keith, we published a second on TALENs and a third on Cas9, which was a collaboration with Jennifer Doudna's lab, our first with her lab. And so our early foray into gene editing was really in trying to achieve a deep understanding of their DNA specificities of these agents and how to improve them. And in some cases, we were able to use the knowledge we gleaned from these unbiased specificity profiling approaches to improve the specificity of these programmable DNA binding domains. So we started, in other words, by simply trying to understand these agents before we ended up engineering and eventually creating our own.
Cool. Yeah, so I was going to say, I think you've highlighted, so when I was first introduced to these zinc finger nucleases, the major, the big problem with it is trying to identify a way of understanding the interaction of the proteins with DNA and how that specificity is achieved, as you mentioned, as by having that information, you can then, in a way, kind of reprogram those nucleases so that they can recognize other sites of interest. And so that's kind of where I guess CRISPR comes in. And instead of using a protein DNA interaction, it uses an RNA DNA interaction, which we understand a lot better in terms of being able to reprogram RNA sequences. And so it gave much more control over the specificity of targeting the editing complex. And so when I first learned about CRISPR, I think I had a kind of wow moment. I definitely remember the moment when I read either the paper I watched Jennifer Doudna talk about it on a TED Talk. I think, I was a bit late to learning about CRISPR technologies. And so I guess, do you remember how it felt when you first learned about CRISPR?
Yeah, it was a kind of aha moment. I think the very first time I started reading CRISPR papers was after hearing about Fanjan's one of Fanjan's talks or papers, where a colleague said, "Hey, you should really check out this area. It seems like it's getting pretty interesting." I remember having a similar aha moment where.
Want to pass your certification exams first time? At Exam Labs, we've got you covered. Looking for a tech role? Don't waste hundreds of hours learning. Get all the answers verified by IT experts. Over 1,000 certification exams, including Cisco CCNA, Amazon AWS, PMP, Microsoft Azure, and much more. With a 98.4% pass rate, you'll have your dream job in no time. Sign up for free now at Exam Labs and start changing your life today.
나랑 결혼해 줄래? 빨리 껴봐.
프로포즈 성공.
네, 안녕하세요.
에너지 풀 충전 가자. 레고 죽어.
요즘 생생한 추억 남기려고 이 동영상 많이 찍잖아요.
이런 분들을 위해 갤럭시 S21이 영상 천재가 되었습니다.
영상 천재 첫 번째 포인트, 바로 디렉터스 뷰입니다.
S21의 전면 카메라와 후면 카메라로 앞과 뒤 동시에 촬영해 볼 수 있어요.
디렉터스 뷰는 카메라 앱에서 더보기, 디렉터스 뷰를 누르면 되는데요. 디렉터스 뷰에서 우상단 화면 선택 버튼을 누르면 촬영 스타일에 따라 양면 분할이나 영상 속 작은 화면으로 선택해서 나를 찍을 수가 있습니다. 또 영상 촬영을 하면서 상황에 맞게 후면에 3개의 화각으로 선택을 할 수 있는데요. 여기 보이는 썸네일로 최적의 화각을 찾아 촬영 중에도 확대, 축소할 수 있어요.
누구를 찍어주면서 동시에 내 모습도 찍히게 되니까 마치 함께 있는 것 같겠는데요? 지금 댓글 보니까 활용 방법이 많이 올라오고 있는데요.
네, 그렇네요.
분할 화면을 이용해서 연인끼리 마주보고 있는 것처럼 영상 찍는 거. 이거 재밌겠는데요? 영상 천재 두 번째 포인트. 싱글 테이크입니다.
움직임이 많아서 예측 불가인 아이들이나 반려견을 키우시는 분들, 이거 꼭 보셔야 됩니다.
단 한 번의 촬영으로 여러 개의 사진과 동영상을 얻을 수 있거든요.
베스트 컷, 필터 사진, 포토 콜라주, 패스트 포워드 영상, 하이라이트 영상. 아이고, 숨차. 저 한번 찍어봐 주시겠어요?
좋습니다.
자.
오, 다양한 사진과 동영상이 차라라라 나오는데요.
저는 찍은 영상을 알아서 AI로 편집해준 다이나믹 비디오가 가장 인상적이었습니다.
오, 저는요 하이라이트 영상이 재밌는데요.
We had been working quite a bit with zinc finger nucleases and talons by then. I think there's no question that the main barrier to zinc finger adoption as a more broadly used biological tool is that there is not really a simple lookup table you can use to assemble effective zinc finger arrays. You can make reasonable candidates that way, but to get them really good, you need to basically do a zinc finger library in vitro selection and a lot of optimization and to have a lot of know-how. And there are a handful of people in this world who can actually do that. And then, of course, tail arrays, I think, became more popular quite quickly because they were actually more or less de novo designable using the lookup table that various scientists, including Bogdanove, uncovered. So when CRISPR came on the scene and the lookup table was basically Watson-Crick-based pairing, I think it did get me very excited that perhaps this was the moment that addressing the genome in this digital programmable way would finally be realized, not just for a company or for a handful of academics with specialized expertise, but maybe for the general life sciences. So it was really exciting. I think nobody back then could have predicted just how democratized it would end up becoming, but it was the start of one of the great revolutions in the life sciences, something that does not come along very frequently. It might come along once a generation or so.
I completely agree. CRISPR has definitely revolutionized, as you say, not only potentially the way we can treat different genetic diseases, but also just as a research tool, with CRISPR screens to investigate and understand how the cells are working. But like with CRISPR-Cas9, the canonical system, in addition to zinc finger nucleases and talons, the way that they work is by inducing double-stranded breaks. And so double-stranded breaks are quite deleterious for a cell. It's a major form of damage for a cell. And it's had raised concerns in different papers about inducing the DNA damage response in terms of the p53 mediated response. And so there's concerns over not only basically the safety of having an editing machinery that induces a double-stranded break. And so that's led to finding solutions that edit without having a double-stranded break that might be potentially safer in case there was any off-target editing going on. And so was that why you ended up discovering base editing, or how did base editing come about? Could you explain a bit about that?
Yeah, it's a great question. So as I think you just gave a great concise summary of double-strand breaks and their role in gene editing. It really began with Maria Jason's seminal discovery that a double-strand break can stimulate increased rates of homologous recombination at the cut site. And because the use of the connection between double-stranded breaks, which is cutting a chromosome into two pieces, And gene editing really began with Maria Jason and others discovering the relationship between double-stranded breaks and homologous recombination products. I think there was great hope early on in the gene editing field's history that being able to make targeted double-stranded breaks with these programmable nucleases, whether they're zinc finger nucleases or TALENs or CRISPR-Cas9, would provide a way to do precise gene correction. Unfortunately, we now know that this process of homology-directed repair, or HDR, as it's called, which is the way that a double-stranded break can be turned into a precise edit, is unfortunately not general. And virtually no therapeutically relevant cell types support HDR with an efficiency that would be considered useful at least for therapeutic applications. As a research tool, it is widely used, and in vitro, under a variety of conditions, you can find cells that support HDR. But, perhaps tellingly, I think when I checked last month, so February 2021, somebody asked me, how many CRISPR clinical trials are there right now? And the answer is, so I looked it up, there's 43 of them. How many of them are trying to use a double-stranded break to induce precise gene repair through homology-directed repair? The answer is 0. All of the therapeutic trials using CRISPR right now are using CRISPR to disrupt genes, because the branch of fate that happens once you make a double-stranded cut in a chromosome is predominantly, in most cell types, to lead to gene disruption through indel formation, meaning the cell is desperately trying to get the ends of the broken chromosome back together. And in the process, it will often get the ends together perfectly, but then the nuclease can just chop up the site again, and you go back to the broken chromosome. So every now and then, the act of joining the ends back together makes a mistake, and those mistakes slow down the process of subsequent cutting. So you see an accumulation of those dead end products, collectively called indels, because they have insertions and deletions. It's mostly deletions. It should be called del-ins. It's just more awkward to say, I suppose. And so, to be clear, making indels in a targeted gene can be quite useful, but you have to get kind of lucky if that is your therapeutic strategy. One really important example where where biology did provide that luck is sickle cell anemia and beta thalassemia, some blood diseases caused by
나 강지영, 국내파 영어 학습 경력 약 20년. 피땀 섞인 노력으로 영어 스펙은 그 누구보다 화려하다고 자부한다.
So, what do you think of this idea?
homozygous mutations in the adult hemoglobin gene called beta globin. You can compensate for those mutations, not by fixing directly the beta globin genes. I mean, you could do that in principle. That's just harder to do it with a nuclease for the reasons we just described. But instead, you can use gene disruption to rescue some of those diseases by disrupting a gene that is involved in the silencing of fetal hemoglobin. So, as I'm sure you know, and your audience is probably becoming increasingly familiar with, we're born with redundant sets of hemoglobin genes. We have a fetal hemoglobin set of genes that is responsible for most of our hemoglobin when we are gestating as fetuses. And then around the time of birth, it begins to be silenced, and adult hemoglobin gene expression begins to be turned on instead. So researchers have identified many of the genes involved in silencing that fetal hemoglobin. And if you can disrupt those genes, then you can actually turn back on fetal hemoglobin. And almost everybody who has defective beta globin genes has healthy fetal hemoglobin genes. So it's probably a reasonably strong selection during embryogenesis. So even if you lose the genetic lottery and you inherit or are born with 2 defective adult beta globin genes, you can still lead a normal life if you have mutations that disrupt silencing of fetal hemoglobin. A very lucky number of people, a small, very lucky number of, well, you could argue they're lucky and unlucky, were born with all of those mutations. They have mutations in their adult hemoglobin that should give them serious blood diseases, but they also have mutations in the genes that are necessary to silence fetal hemoglobin, so they end up being okay. And so a bunch of really smart scientists and companies have exploited this observation to use CRISPR-Cas9 or other double-strand break inducing programmable nucleases to disrupt the genes that silence fetal hemoglobin. So that's an example of how gene disruption can actually benefit a patient. But the reason I said you have to get kind